Hello, and thank you for joining this HCP Live peer exchange titled Sickle Cell Disease, Diagnosis, Screening, and Treatment. Sickle cell disease is an inherited disease that affects red blood cells with an abnormal version of hemoglobin. The disease is associated with acute and chronic complications and shortened lifespans. In this HCP Live peer exchange, I'm joined by a panel of my colleagues, all experts in the field of hematology. And together, we will discuss the challenges associated with the diagnosis and the pharmacologic options for the treatment of sickle cell disease. I'm Dr. Peter Salgo. I'm a professor of medicine and anesthesiology at Columbia University College of Physicians and Surgeons. I'm associate director of surgical intensive care at New York Presbyterian Hospital in New York City. And with me today on our distinguished panel are Dr. Bari Andemariam, associate professor of medicine and director of New England Sickle Cell Institute at UConn Health in Farmington, Connecticut. Dr. Jane Hankins, associate member in the Department of Hematology at St. Jude Children's Research Hospital in Memphis, Tennessee. Dr. Sophie Landskron, director of the Sickle Cell Center for Adults and associate professor of medicine at the Johns Hopkins Hospital in Baltimore, Maryland. And Dr. Elliot Vichinsky, chief of hematology oncology, professor of pediatrics in the School of Medicine at the University of California, San Francisco in San Francisco, California. Thank you all so much for being here. It's a delight to meet you. Some of you I've met before. Yeah. This is great. So why don't we start with the real basics here. Somebody want to define sickle cell disease for me? Who wants to start?
Well, I'll start.
Brave of you. Go right ahead.
I think it's important for people to recognize that sickle cell disease really is the most common hereditary blood disorder worldwide.
The most common?
Yeah.
Okay.
Yeah. And it affects 300,000 babies per year that are born across the globe. And it's estimated that here in the United States that there are 100,000 Americans living with sickle cell disease.
I want to pause just for a minute because those numbers are large.
They're very large.
And I think that there's a tendency for folks who don't live in endemic areas and parts of our country to underestimate how big a problem this disease is, don't you think?
In our country?
Yes.
I agree with you. So if you look at, I also think it's important to realize that although sickle cell disease is most common in individuals of African origin and Latino background in this country, that the face of sickle cell disease has told us that just about everybody can be affected.
Really?
Yes, I think all of us will have examples in our practices of individuals who are neither black or brown. It's also seen in individuals from the Mediterranean, from the Saudi Arabian Peninsula, and also in Southeast Asia. So it's not just a black and brown disease, although in this country that's predominantly what we see.
I want to take your point to an even broader interpretation. You don't have to have sickle cell, or you don't have to have an immediate relative with sickle cell to be affected by it. As a nation, with that many folks with this problem, I think we're all affected. Is that a fair statement?
Well, I would like the country to feel that way. We clearly are. But sickle cell disease has been a disease that has suffered from discrimination because of the ethnicities that it involves in. But it clearly affects the country. And there are many people who are carriers or have other types of sickle cell disease in this country that are not in one particular ethnic group.
Well, let's get some more basic facts on the table, all right? Genetic underpinning of this disease, the difference between the trait and sickle cell anemia.
Sure. Yeah, so I was just going to add that sickle cell disease is considered a rare disease, but it's not really, because when you look at the distribution of sickle cell disease, it's kind of concentrated in certain urban areas. But sickle cell disease is caused by a genetic mutation. It's a single point mutation in the gene that makes the beta globin. And the beta globin is what produces part of our hemoglobin, which is the protein that carries oxygen. So when you mess up that beta hemoglobin, when it binds with the other globin, which is the alpha globin, it creates this hemoglobin that really doesn't behave very well. And then it likes to bind with other abnormal hemoglobins that we call sickle hemoglobin, and that clogs the circulation or causes vaso-occlusion.
All right, so let's go one step further. That's the genetics. How does this work? In other words, what is the genetic and epigenetic factors that lead to the actual sickling?
Before we get there, I'd like to just add one thing when we started to this. Many of the types of sickle cell disease are not anemic, and they have different physical characteristics, so they often go undiagnosed. So the disease itself is really about the pathology of the cells adhering and the problems they have, not anemia. So I think that's important.
Well, we're going to get to there. pretty soon, actually, right after this. So how do the cells, I guess, what makes them sickle? We have these hemoglobins, they're abnormal. What actually goes on? Do we know why they sickle like that? Do you know?
Oh, well, yes. I think sickle cell disease is probably one of the best studied molecular diseases historically, and there's been a lot of work into understanding of that. And basically, hemoglobin is made up of 2 alpha and 2 beta chains, and when you're in utero, you make mainly alpha chains and gamma chains, and after birth or around them, you start making the adult hemoglobin A. These people have a mutation that converts that to the sickle hemoglobin. Okay. And then, since they don't have symptoms in utero, but with this point mutation, it results in the hemoglobin being okay, relatively okay when the cell is oxygenated, but when oxygen drops, the polymers or the molecules inside the cell that are kind of floating around suddenly become rigid, and they stick together and they deform the cell, and they make the cell have that sickle shape, but in addition, the membrane dramatically changes, and the inside of the membrane flips to the outside and exposes a great number of phospholipids and changes that induce remarkable explosion of inflammatory and thrombotic products. And it destroys the membrane, so the cell basically starts falling apart very early.
I just wanted to go back to the genetics for a moment and just remind people that there are several different types of sickle cell disease. So people can be homozygous for the hemoglobin S mutation and have SS disease, but then there's SC disease and S beta plus thal. And as we talk about therapies, it's going to matter what kind of genotype patients have.
That's why they pay you the big bucks. You know that? You know these things.